The goal of gene editing is to change the building blocks of genes in living things, in order to better understand how these genes work, and to facilitate the treatment of inherited and acquired disorders. Genome editing can be used to correct, introduce, or delete almost any DNA sequence in a variety of cell types and organisms. While methods to alter DNA have been around for a while, new approaches have made gene editing quicker, less expensive, and more effective. CRISPR-Cas9 is the most widely used gene editing system today. This system consists of two parts: a tiny strand of guide RNA designed to look for a specific DNA sequence, and an enzyme, Cas9, that can cut through a double strand of DNA. CRISPR stands for clustered regularly interspaced short palindromic repeats, which are segments with a certain DNA structure that are prevalent in bacteria and archaea. In 2020, two scientists, Jennifer Doudna and Emmanuelle Charpentier, shared the Nobel Prize in Chemistry, for their discovery of the revolutionary CRISPR-Cas9 gene editing technique. With CRISPR, DNA can now be altered at precise locations and at multiple sites, involving anywhere from single base pairs to large pieces of chromosomes. Once inside a cell, the CRISPR system searches for the DNA it has been trained to look for. The CRISPR-seeking tool subsequently locates and binds to the target DNA. Both DNA strands are cut by the Cas9 enzyme. New DNA segments can then be inserted into the cell. The cell automatically incorporates the new DNA into the gap made by Cas9, when it repairs the broken DNA. Gene editing applications in research. 1. Creating tailor-made organisms, to study human diseases. 2. Improving strains of agricultural crops. 3. Altering vector populations, to help interfere with the transmission of infectious diseases. 4. Modifying yeast cells to make biofuels. In clinical settings, gene editing could be used to treat genetic diseases. However, until reliable exact genomic modifications can be achieved, without generating unwanted mutations, human embryos with modified genomes shouldn't be used to induce a pregnancy. Heritable genome alterations can be passed down to future generations, thus posing a variety of moral, ethical, and social concerns, in addition to scientific and medical uncertainties. Before any nation decides whether to legalize the clinical use of heritable human genome editing, with the aim of establishing a pregnancy, much social and scientific debate is required. Ideally, we don't want a world in which there is a possibility of creating designer babies, for purposes of enhanced capabilities, for example in sports. Initial uses of heritable human genome editing should therefore be restricted to the avoidance of serious monogenic disorders, which are caused by the mutation of one or both copies of a single gene. Examples of such diseases are cystic fibrosis, thalassemia, and sickle cell anemia.